Hello everyone, and a massive welcome back to MooTechie. We're so glad you're joining us for another fascinating topic. Today, we're diving into the world of meat, exploring the intricate journey from the farm to your fork, the unseen microbes that hitch a ride, and the constant battle against spoilage to ensure the meat on your table is safe and high quality. This is a story of biology, chemistry, and careful handling, so let's get started. Now, let's begin our presentation. Microbiology of meat and meat products, sources of contamination, spoilage and prevention. Our journey begins with production. The process involves transporting the animals, holding them, and then slaughtering. For animals like cattle, sheep, goats and horses, the key steps are skinning, which is a critical control point or CCP2, eviscerating, also a CCP2, washing, chilling, which is a CCP1, transporting, a CCP2, and finally, cutting, boning and packaging. For pigs, the process is slightly different. It starts with scalding, a CCP2, then dehairing, singeing, a CCP2, polishing, eviscerating, a CCP2, chilling, a CCP1, transporting, a CCP2, and finally, cutting, boning and packaging. Now, what is meat made of? Let's look at its proximate composition. Water makes up the majority at 75 to 80%. Protein is next at 18 to 22%. Carbohydrate is less than 1%. Fat can range from 3 to 13%. And there are only traces of vitamins and minerals. In a healthy and physiologically normal animal, the internal organs that have no direct contact with the exterior are virtually sterile. However, the operations of slaughter and dressing have a significant potential to introduce bacteria to the blood, tissues and organs. These microorganisms are usually a mixed flora from the environment and fecal origin. Alarmingly, they can include pathogens like Salmonella, Campylobacter, E. coli, and other food poisoning organisms. Furthermore, specific pathogens may already be present in organs or tissues due to a generalized infection in the animal at the time of slaughter. Assessing the fitness of meat requires 3 stages: ante-mortem inspection, gross post-mortem inspection, and if necessary, further laboratory tests. Now let's talk about the inevitable processes of meat decomposition and spoilage. Decomposition is the process where organic matter is broken down into a simpler form. In meat, this primarily affects protein but also fats and carbohydrates. This breakdown imparts off odors, off flavors, and leads to slime formation. Bacteria, molds, and yeasts are the main culprits, reducing the meat into simpler, often foul-smelling chemical substances. All natural foods remain fresh for only a comparatively short time before bacteria take action. Foods are rapidly colonized, leading to decomposition through enzyme action and oxidation. Enzymes present in all living cells catalyze the chemical reactions in the cells. The process of autolysis or self-degradation is brought about by these enzymes at a rate that varies in different tissues. It is highest in tissues that synthesize large amounts of protein and have high water content, like the gastrointestinal mucosa, testes, pancreas, and adrenals. The shelf life of any food depends on its structure, pH, composition, water content, the presence of bacteria, damage, and storage conditions. A critical point is that meat from fatigued animals spoils faster. The pH of meat from these tired animals after rigor mortis is around 6.5, rather than the lower normal value of around 5.6 in a rested animal. This lower pH of 5.6 is crucial because it slows bacterial growth as it's outside their optimal range, thus protecting the meat. The main spoilage bacteria in meat belong to several genera. Fresh meats can be initially contaminated from many sources: soil, dust, feces, water, equipment, and personnel. It was once thought that the flesh of healthy animals was sterile at slaughter, but we now know it can harbor organisms, mostly gram-positive mesophiles. Depending on the bacteria present, foodborne disease, spoilage or both may result, especially with substandard handling. Let's meet the spoilage organisms, starting with the gram-positive ones. Bruchothrix grows in both aerobic and anaerobic conditions. Its optimum temperature is 20 to 25 degrees Celsius, but it can grow at temperatures as low as 0 degrees. Carnobacterium grows anaerobically in high CO2. Some can produce antimicrobial peptides called bacteriocins. Micrococcus species can be salt-tolerant, heat-resistant, or cold-loving. They cause spoilage in salted and chilled meats. Staphylococcus species are also salt-tolerant. While Staph albus causes spoilage, Staph aureus is a major cause of food poisoning. Streptococcus species, like *str. faecalis, have a wide growth temperature range and some salt tolerance. Lactobacillus are mainly mesophilic. They can grow at a water activity as low as 0.91 and at a pH less than 4.5. Leuconostoc are lactic acid bacteria that can produce slimes and flavors from diacetyl. They can also produce hydrogen peroxide, which gives spoiled meat a green discoloration. Bacillus species are biochemically very active. Some can cause flat sours in canned meats. Now for the gram-negative organisms. Pseudomonas are the predominant bacteria in spoiled meat. They are widely distributed and grow well in protein foods, producing slime, pigments, and odors. Flavobacterium produce pigmented colonies that cause discoloration. Acinetobacter can oxidize ethanol to acetic acid. Achromobacter are similar to Pseudomonas and also form slime. Helobacterium are salt-loving and spoil high-salt meats. Moraxella are sometimes classified with Acinetobacter. The enterobacteria are abundant in soil and intestines. E. coli is indicative of fecal pollution and can cause spoilage. The verotoxic strain, E. coli O157H7 is a major food poisoning bacterium. This group also includes pathogens like Salmonella and Shigella. On chilled fresh meats, these groups are responsible for slime formation. The time for slime to develop is directly related to the initial number of organisms on the carcass. Therefore, efficient hygiene at slaughter during dressing, refrigeration, storage, and transportation is absolutely critical. Chilling doesn't prevent spoilage, as some organisms can grow at around -7 degrees Celsius. However, temperatures below 2 degrees Celsius will delay slime formation. The spoilage process starts with the breakdown of carbohydrates. As time passes, protein molecules are broken down by acids, alkalis, enzymes, and bacteria. Putrefactive bacteria break proteins down into simpler and simpler compounds, eventually producing foul-smelling gases like hydrogen sulfide and ammonia. The recognized signs of decomposition are changes in color to gray, yellow, or green, a softening of the tissue, a repulsive odor, and an alkaline reaction. After slaughter, decomposition starts on the surfaces exposed to air. Aerobic bacteria like Pseudomonas initiate the process, consuming oxygen and creating conditions for anaerobes like *Cl. Sporogenous to grow in deeper tissues. A condition known as heated beef or sourside occurs when carcasses can't cool rapidly enough. A more significant consequence of slow cooling is that it destroys enzymes needed for aging, impacting meat tenderness. In animals that have not been promptly eviscerated, decomposition occurs rapidly from the inside out, often indicated by a greenish hue on the kidney fat. In pigs, delayed evisceration can cause a greenness of abdominal fat in just 12 hours, a condition known as sulfiding or pseudomelanosis. It's vital to remember that while spoilage organisms reveal themselves through off-colors and odors, most food poisoning bacteria give no such warning. Now let's talk about the decomposition of fat, known as rancidification. This can follow oxidative, hydrolytic, or microbial pathways. Oxidative rancidity is the most common type and doesn't require microbes. Unsaturated fats are particularly susceptible, and this process can continue even at low temperatures. Beef and mutton fats are relatively resistant, but bacon fat is much more susceptible, especially after curing, as the salts can accelerate oxidation. A serious condition is bone taint, a deep-seated spoilage near the hip or shoulder joint, caused by putrefactive bacteria, often from the animal's gut. It can be reduced by rapid cooling and avoiding contamination. Taint in hams, also called souring, is attributed to bacteria like *Cl. Sporogenous. Sometimes, meat can even show phosphorescence, caused by bacteria like Pseudomonas phosphorescens, making the meat appear luminous in the dark. Molds are another common issue, appearing as fluffy growths. They thrive in moist conditions and can grow at low temperatures. Common types on meat include black spot, caused by Cladosporium herbarum, white spot, caused by Sporotrichum, and whiskers, caused by Thamnidium and Mucor. Bluish-green molds from the genus Penicillium are also frequent. Finally, how do we assess decomposition? Various chemical, bacteriological, and physical tests exist, but none can fully substitute for the organoleptic inspection by an experienced inspector. Chemical tests might detect ammonia or volatile bases. Bacteriological tests count bacteria, but this doesn't always correlate with the sensory spoilage. Measuring the pH of deep muscle is also a useful indicator. An interesting historical method is chemiluminescence, which measures the light emitted by deteriorating fats to gauge freshness. And that brings us to the end of our presentation. Thank you. We've traveled the entire path of meat production, from the slaughterhouse to the shop, uncovering the vast microbial ecosystem that challenges its preservation. We've seen how factors like pH, temperature, and hygiene are paramount in controlling spoilage and ensuring safety. Understanding this complex interplay between meat and microbes is fundamental for everyone involved in bringing this essential food to our tables. Thank you for your time and for your commitment to food science. We look forward to sharing more knowledge with you in the next session.